Okay, so welcome back to this next video in which we are discussing the reactivation of replication forks by homologous recombination. Okay, so we're discussing replication fork reactivation, or RFR. So we've now discussed the basic pathway which is going to occur. What we want to see is the actual proteins that are going to be involved in guiding this pathway, okay, of homologous recombination. Okay, and we're going to see this for Escherichia coli. So I want to stress again that you find this in all cells. You know, this is a repair mechanism that human cells will most definitely have, but the proteins have been far better characterized for E. coli. So we're going to talk about the proteins within Escherichia coli, which is a bacterium. Okay, right, so we've now discussed the pathway in full again, but we'll do it in the most detail now, so we're gradually moving up the scales, basically. Okay, right, so once again, let's start off with our chromosome, and I think I'll try and use the same colours as before. Okay, so we'll start off with our chromosome here, which has these two strands. Here is the turquoise strand, and here is the blue strand here. Okay, and we'll say that the turquoise strand's 5' end is this side, and its 3' end is this side, and the blue strand's 3' end is this side, and the blue strand's 5' end is this side. Okay, right, so we've then decided to replicate these chromosomes, okay, well, this chromosome rather, okay, and we're opening a replication fork at this side over here. So here's our turquoise strand. Okay, here is our blue strand as well, so the two of them have been pulled apart at this side, but they're still bound in a double strand together for the rest of the length. Okay, then we've got our two new strands being synthesized. So here is our new red strand, which is complementary to the turquoise strand, and then the other strand was in orange, which is complementary to this blue strand here. Okay, now we've then got a problem. We've got this single strand break in the turquoise strand just ahead of the replication fork here. So what's going to happen is when the replication fork actually meets that single strand break, you're going to get a broken replication fork. Okay, the turquoise strand is going to completely detach off from the blue strand and the rest of the turquoise strand, and the red strand will now be fully complementary to that little fragment that we've got there. Okay, then we will still have a complete chromosome here, because here is the rest of that turquoise strand. The portion that we've lost in the form of this turquoise fragment up here has been replaced by this orange strand here, and then we've got the blue strand, which is still completely intact here. Okay, right, so now what we remember occurs is an enzyme is going to come and bind up here, okay, at this 5' at 3' end up here. Okay, so this is the 5' end of the turquoise strand and the 3' end of the red strand here. Okay, so we're going to get a complex binding at this free end of the DNA. And remember, this side will not be free. This is the actual end of the chromosome over here. So this is different. This will have other structures on it that will stop the complex from binding there. This is the free broken end. Okay, so it's going to come and associate at this side. Now, this enzyme complex that's going to associate is what's known as the REC-BCD complex. Okay, and it contains three proteins called REC-B, REC-C, and REC-D. Okay, so that's why it's called the REC-BCD complex. Okay, so let's draw a picture of this. So I'll draw the REC-BCD complex like so. Okay, so it's made up of these three separate proteins here. Okay, REC-B is at the bottom here. Okay, REC-C is going to be in the middle there, and then REC-D is at the top, so let's colour them all in. So we'll have REC-D in yellow here. Oh yeah, that's smudged nastily, never mind. Then we've got REC-C in the middle, and we'll have REC-C in vivid purple here. Okay, and then finally we've also got REC-B, which we'll do in green. Okay, so this is our RecBCD complex, and now basically this is going to bind onto our 5', to our end of our broken end of this little fragment of DNA here. Okay, like so. Here comes our strand of DNA. Okay, so this is the red portion here, and this is the turquoise portion here. And what it's going to do is it's going to work its way along the DNA, and it's going to split the two strands apart, and it's going to cut these two strands up. Okay, so it's going to be working its way along like so, and it's going to be cutting up the turquoise strand, cutting up the red strand, and it's going to be working its way along, cutting both of them up, basically. Okay, so this is this initial trimming that both sides get, until, remember what happens, it's going to reach that chi sequence here. At least in E. coli, it's going to reach this chi sequence, which I'll remind you is a G, C, T, G, G, T, G, C sequence of 8 organic bases, and this is the 5' end, and this is the 3' end. Okay, so this will occur on this red strand here. Okay, and when it finds that, what's going to happen is this RecC protein here is going to bind to that chi sequence, okay, and it's going to stop the RecBCD complex from breaking up the red strand anymore, which, remember, is significant because the red strand has this 3' end at the end of the BCD complex. Okay, right, so let's draw this now, okay. So what's going to happen, we'll start by redrawing our RecBCD complex. Okay, so here is this complex of these 3 proteins. We've got RecB here, RecC here, and RecD here, and we'll colour them in again. So we'll have RecB in green here, RecC in vivid purple here, okay, and then we'll have RecD in yellow here. Okay, then it's worked its way along the DNA until it's got to this chi sequence. And now what's going to happen is this RecC protein is going to bind to that chi sequence. So here is the chi sequence here in red. Now, the RecBCD complex is going to stop working its way along the red strand now, but it's going to continue working its way along the green strand. So what's going to happen is the green strand is going to continue being moved through here, and that means that you're going to get a loop of red strand building up like so. Okay, whilst you're working your way along the green strand still, or the turquoise strand rather, and breaking it down. Okay, that's how you build up this 3' extension. Okay, so it produces this overhang, and it will continue on for a certain amount of time, and then it will decide that the 3' overhang is large enough now, and it will stop, basically. Okay, then what it's going to do is it's going to recruit proteins onto this 3' extension that we have that overhangs the turquoise strand. Okay, and it will start at this side over here. So remember, this red strand, this is the 5' end of the red strand, this is the 3' end of the red strand, this is the 3' end of the turquoise strand, and this is the 5' end of the turquoise strand. So once the RecBCD complex has stopped cutting more of this turquoise strand out, basically, it will start putting on another protein. Okay, so it's going to start loading another protein known as RecA proteins. Okay, and it's going to start loading these onto the 3' extension of this red strand that we have here. Okay, and it will start at the 5' end of the 3' extension. So it's going to start here, putting on a RecA protein there, then it's going to work along and it will put more and more on. Okay, and what it will overall produce then is, to draw this out, here's our turquoise strand, okay, and here's our red strand with its 3' extension here, and we're going to gradually put on RecA proteins starting at this side, and we're going to put more and more of them all over the 3' extension here. So these are all Rec-A proteins, and these are what are going to help with strand invasion. So these are going to help the strand to invade the other complete strand up here. Okay, right, so we're going to load on loads of Rec-A proteins, starting at this side, working our way this side, towards this side, and it's the Rec-BCD complex that does that. Okay, after the Rec-BCD complex is finished, it will then obviously detach off. So once the 3' extension is covered in Rec-A proteins, that's the Rec-BCD complex done. Okay, now let's bring our other strand here, down here, okay, so that we don't forget that. In fact, actually, let's draw this strand out again, because we've made it much bigger than it was shown up here. So I want to now take it back down to a good relative size. Okay, so here is our little red strand now. Okay, so I'm shrinking this picture basically. Here are our Rec-A proteins bound over it. Okay, and here in turquoise is the turquoise strand. Okay, then we've got our other strand here, and unfortunately I've used the same colour for Rec-A as I have for this strand, but never mind. Here's the orange strand. Here in turquoise is the rest of the turquoise strand. Okay, and then here in blue, that's the one strand that's still intact. Okay, so the next step is that we're going to get strand invasion. So remember what happens in strand invasion. This 3' extension is going to invade and put this other strand here. And it's going to detach this orange strand here from the blue strand here. And it instead is going to bind that orange strand to the red strand. Okay, so this begins once again at this 5' end of the red strand. So let me show you this. So I'm going to put in a few more steps than I showed when we were looking at that bigger picture. Okay, so here is our turquoise little fragment, which isn't doing anything yet. We'll come back to him later. Okay, then here is our red fragment. Okay, now what we're going to start off with is we're going to move our orange fragment out like so, and we're going to have a little portion that is now attached to the red strand, but then the rest is going to be attached to the to the blue strand. Okay, so we have now detached our orange strand a little bit. Okay, and notice the side of the 3' extension that we are working with at the moment. We've attached this orange strand firstly to the portion at the 5' end of the red strand here. So remember, this is the 3' end, this is the 5' end. So we've attached the orange strand to the 5' end of the 3' extension. Okay, now meanwhile, we're still going to have Rec-A proteins attached to the rest of this 3' extension that isn't yet bound to the orange strand. So as you bind the orange strand to the red strand, or the 3' extension of the red strand, the Rec-A proteins are going to come off. Okay, so you'll start at the 5' end of the 3' extension, so this side over here, okay, and you'll take the Rec-A proteins off there, and then you're gradually going to extend this connection between the orange strand and the red strand. So let me draw that now. Okay, so you're going to extend this connection, so let me draw it. So here is our little turquoise fragment to start off. Here is our red strand here, okay, and now we're extending the connection between the orange strand and this 3' extension so that it now covers the entire thing, and that will gradually pull off these Rec-A proteins. Okay, now I'll draw it back the way I drew it in the previous picture. Oh, whoops, I've forgotten the turquoise strand. We'll have to put that sort of right over here. Okay, and then we've got the blue strand here, going all the way on. Okay, right, so there we have strand invasion. So we've split strand invasion into these two separate steps now, because remember, the Rec-A proteins are removed gradually, they're not all removed at once. Okay, so you take a Rec-A protein off and you extend the connection between the orange strand and the red strand. You take another Rec-A protein off, you extend the connection between the red strand and the orange strand until the entire 3' extension is connected with the orange strand like this. Okay, so strand invasion is now complete. Okay, so what's going to happen next? Well, remember, we are going to now form the holiday intermediate by performing branch migration, okay, and I think I'll try and squash this in here, okay, or maybe I should go over onto the other side. Yeah, I'm going to go over onto the other side because we're going to have to move at some point, so we might as well use this as an opportunity. So let me just draw out the picture that we had on the previous page, because it would be awful for me to now just draw the next picture without the other picture drawn there. Okay, so remember, here is the turquoise strand, here is the red strand here with the 3' extension. Okay, we've now got our orange strand here, and we've got that strand invasion, so the orange strand has now got that connection with the 3' extension of the red strand. Here is the rest of that strand in turquoise over there, and then here is our blue strand here. Okay, right. So what's now going to happen is you're going to get branch migration. So the next process is branch migration. And remember, what happens in branch migration is you're going to peel this turquoise strand away from the red strand, you're going to peel the orange strand away from the blue strand, you're going to transfer the orange strand onto the red strand, and then you're going to move this portion of the turquoise strand onto the blue strand to form that holiday intermediate. Okay, so we'll start off with this tiny little portion of the turquoise strand that's still attached to the red strand here. Okay, and then we've got the orange strand here, which has now got this much bigger connection with the red strand, like so. Okay, then we've got in turquoise here the continuation of that strand. Okay, and in blue here is our complementary strand, which will be attached over here to the tail of this orange and turquoise strand. But now, of course, we've attached the other portion of the turquoise strand down to this blue strand down here. Okay, and the name for this structure that we've created here is a holiday intermediate. Okay, right. So remember, this is our nice picture as far as understanding is concerned, but if we're going to discuss what's actually happening in the E. coli bacterium, we need to draw that other picture, which is how it actually looks within the cell. Okay, so let's start by drawing our big strand, and then we'll put in our smaller strand. So here is our orange strand here. Okay, and remember, it's opening up here, which is this opened up portion here, and then it's coming down here. It's becoming the turquoise strand once again down here, let's say. Okay, I'll bring this up a little bit. And then we've also got that blue strand, and they come apart at this portion, and then they reconverge together over here. Okay, then we're also bringing in this other strand, which is the red and turquoise strand. Okay, so let's say we've got the red strand here. Okay, and this has now got this connection with the orange strand here, and we've now got our turquoise strand, which is here, which is shorter than the red strand, remember, which has got this connection with the blue strand. Okay, so this is what a holiday intermediate actually looks like in a cell. Now, proteins are going to assemble around this. 4 proteins assemble around this holiday intermediate, and by the way, this is why it's called a holiday junction. This structure that we've got here is what's called a holiday junction. Okay, so that's where this picture is useful for understanding why this is called a holiday junction, because that looks exactly like a junction in roads now. Okay, and 4 proteins are going to bind to this holiday junction here, and these 4 proteins are identical. Okay, so here we have them in vivid purple here, and these 4 proteins, 1, 2, 3, 4, are all known as RUV-A proteins. So all of these pink triangles, they are RUV-A proteins. Okay, now, in this holiday junction we have 2 inputs, okay, so these are both input strands. So we're inputting this, and we're inputting this. So you can imagine we are shoving these 2 portions in, and the outputs are then coming out here. So this is then an output, and this is then an output. Okay, so let me just colour that up. So actually, I don't think I will colour that up because I need to draw other things here. Okay, so then what's going to happen on top of the tetramer of RUV-A proteins, you're then going to get a ring assembling around these output strands here. Okay, so a ring that will assemble around these outputs, and this ring consists of 6 separate proteins, 1, 2, 3, 4, 5, 6. Okay, and you're also going to get one of these rings assembling around this output strand here. Okay, I'm really regretting drawing those little output bits now. Okay, there we go. So what colour should I colour these in? Turquoise, I think, if that's not going to be too confusing. So here is our ring of 6 proteins that is assembled around here, and these 6 proteins that have assembled here, these are RUV-B proteins. Okay, so we've got these rings, these hexameric rings of RUV-B proteins. Okay, so on the holiday junction here, we are assembling what's known as the RUV-AB complex. Okay, right. Now, what's the significance of this? Well, these RUV-B hexamers, these are what are known as DNA translocases. Okay, now what does that mean? Well, it means they're going to use ATP, basically, to pull the DNA through them. Okay, so they're basically going to be pulling on these 2 portions, and they're pulling them through. They're pulling the output strands through, and that, if you like, is going to pull the input strands in to produce more of these output strands. Okay, so this is how you're going to be getting the DNA recombining in this way, because these RUV-B hexamers are using ATP to pull these 2 strands apart, and well, pull these 2 strands together, if you like, and they're recombining the 2 input strands into these 2 output strands. Okay, then what are you going to do? Well, let's remind ourselves what happens after we have formed this overlap, like so. So actually, let me just summarize. This RUV-B is responsible for the branch migration, basically. It's what's migrating these branches backwards. It's what's responsible for peeling the orange strand away from the blue strand here and then binding it to the red strand. So it's peeling the red and the turquoise strand apart, it's peeling the orange and the blue strand apart and then recombining them together. Okay, so it's responsible for the branch migration. Now, once we've had enough of branch migration, what do? We cut this strand here, we cut the turquoise strand and we cut the orange strand, and we then say, okay, this portion of the orange strand will go on to this red strand here and we'll religate it with this turquoise fragment here. Then this portion of the turquoise strand here will go onto this blue strand and we'll ligate it with this fragment of the orange strand here. Okay, and remember what that overall then produces is, to draw the final product here, we have our turquoise strand with our orange strand now, so this is this large portion of the orange strand, and then it will have the turquoise strand downstream of that, that's this portion. Then we've got the blue strand here. Okay, and then on that blue strand, what have we got? We've got this orange portion here and then the turquoise portion. Okay, so let me just draw that. So here is that first little orange fragment there, and then we've ligated that to this turquoise strand here. Okay, so that's that portion, and then on this other strand we've got the red strand, and then we can resume replication because we have produced a new replication fork. Okay, so to draw that finally on this picture, what's going to happen is we're going to cut this strand here. Okay, so remember that's analogous to this cut here. So we're cutting this orange strand here. Okay, so we're going to cut there, and then we're also going to cut here. Okay, and that's analogous to this cut here. Okay, then we're going to recombine this orange portion with this turquoise portion down here, and we'll recombine this orange portion with this turquoise portion here. Okay, now, what is the enzyme which actually produces those cuts? Well, it is the RUV-C enzyme. Okay, so RUV-C is the enzyme which will cut the DNA, so this is a special cleavage enzyme. Okay, right, so then let's finally just finish this picture off. So what will happen is the RUV-AB complexes will disassemble, and then what you'll get is you've got your orange strand here, so this is this first orange strand here, then we've got our blue strand here, which will come round like so. Okay. Then we've now attached this orange strand here to this portion of this turquoise strand here, which will come out like here. Okay. Then we've got our red strand here. Okay. So I'll put the red strand here, going off like that. Then we've attached the first portion of this turquoise strand, so this portion here. We've now attached that to the second portion of the orange strand here. Okay, so this will come round like so, right down here, and then it will recombine with this blue portion. And then what we can do is resume the replication fork here. Okay, so that then is how we can use homologous recombination to reactivate replication forks. Okay, and as I said, we have studied the actual proteins that are involved in this in E. coli because that's where it's best understood, but the pathway, the basic principle of the pathway of how you can use homologous recombination to resolve these broken replication forks, that transfers over into human cells.